or some of you might feel that it is very dry and what's happening. So it's a mixed response that we get. But anyways, to start this topic, first we have to address why we have to study these principles of inheritance and variation is the first question. So if you see as a biologist, so one of the best things that we just have in common is that we appreciate the diversity. So what is diversity? The variations that we see in the environment. So we appreciate the different colors of flowers, we appreciate different varieties of birds, we appreciate different varieties of mammals, we love the biodiversity. For all of this, we really enjoy, and we feel very, very lively when we see all these things, right? And in the back of our mind, we just try to have this question, like, how are they so different? How are they so unique? And sometimes, how are they so same? So this is the question that runs in our mind. So one thing that haunted a human mind is who is carrying the characteristics from one generation to another generation, or how the transmission of the characteristics from one generation to another generation occurs. So this was the biggest question for many, many, many centuries. But until and unless it took some versatile minds or different unique minds who came across with solutions, okay, and defined how the things or what things are responsible for the transfer of characteristics from one generation to another generation. Let's see in this chapter and understand why the genetics, why genetics, how the genetics works, or what is the significance of understanding the genetic variability or inheritance, so that we many of our questions on why it didn't happen like that, or why is it like this, why am I not looking similar to similar to that of my parents, or why am I very similar to my father, not similar to my mother. So these questions can be answered very easily with a very simple understanding of concepts of genetics. Okay, so what is genetics? So whenever we talk about genetics, the one of the common things that we share with all the living organisms is the capacity to reproduce. So when we reproduce, what happens is we transfer the characteristics from one generation to another generations. So why does this happen? Why should we transfer the characteristics from one generation to another generations? So people say that, okay, we have a genetics which is quite selfish. All the organisms on our planet Earth are selfish, and our gene is a selfish one. So that's why we call it as a selfish gene. Why it is selfish? So our idea of a living organism is to live forever. Any organism you take, it wants to live forever. Many you might address the fear of death, or if you're not having the fear of death in a second, in another sense. But on a whole, every organism wants to live forever. It wants to be eternal. Even though some might disagree, sir, I don't want to live. It's because you are young. So as you move on, you want to have more years for yourself. But you know that the laws of nature has modified itself, and it has made sure that it lives on forever in through its characteristics. I cannot live in the same physical body forever, but through the transfer of my traits to one generation to another generations, my traits can live forever, and it can be passed down and down and down, and it can get modified and modified, and it can become better and better organisms from one generation to another generation. It is such a beautiful poetry to understand how the genetics and how the living organisms came up with this plan of transfer of characteristics from one generation to another generation, isn't it? Yeah. So if we see that the result of reproduction was not just to make offsprings, it was to make the copies, exact or similar copies of the parents. So to make the copies, somebody has to take this information, so much information from the parents all the way till the offsprings. And not just take this information from the parents to the offsprings, the parents who are living in a particular environment go through the experiences of life and understand what was lacking in them, how they were not able to adapt, what conditions are going to come, or what are the competitions they are going to face, what is their enemy, what is their friend, what is their food, how is it changing. All these life experiences will add up and give the proper guidance to the next generation to ensure that it has more adaptable nature, more intelligent nature, more agile, and more evolving, I can tell. So this gives the organisms the ability to continue from one generation to another generation, and it will give the organism the ability to adjust and adapt. Correct, right? So because the conditions are not always same. So anyways, so the resulting offsprings are not resembling, or sometimes it is resembling to the parents. Anyways, so some of the terms that we have to be aware before understanding the heredity is, there are few terms, we'll talk about it. Okay. First one is, what is genetics? So genetics is a branch of life science. What we do here is we study how the heredity and variation occurs in living organisms. So we have two terms here, what is heredity and what is variation. So as I mentioned that we study in living organisms how the transfer of characteristics takes place from one generation to another generation. So this is heredity. And what is variation? Variation means, why am I not resembling my parents exactly? So that is variation. So in this, we address two questions which are quite opposite. Heredity is transfer of characteristics from one generation to another generation, or from parents to offsprings. So that means I should have identical characteristics to that of my parents. Whatever the characteristics of my parents were there, I should carry them to my generation. I should express them. And so does my child has to express, and their children and their children. So this is called as heredity, where we take these characteristics, express and pass on. So this is called as heredity. Whereas when you come to variation, variation is quite opposite. Even though if you see, we have an offspring, okay, coming from the same parents. Let's say that a parent, a mother and a father, give birth to two children, one male and one female. Now you can see that either one of them, neither one of them, one will be resembling their parents, and the one, another one will not at all resemble neither mother nor father. Let's say that we have a daughter and we have a son. The daughter, what she will do, she will be very similar to the mother, very similar to mother. The facial characteristics, that the skin tone, or the texture of the hair, the intelligent quotient, the height of the girl would be very, very similar to that of the mother. Whereas the son, he would be taller than the parents, he would be dark skinned, okay, and his intelligent quotient might be low or very high. So very, very non-parental. So whenever you have a characteristics that does not resemble with the parents, so these are called as variations. So are variations something not hereditary? Let's observe that. Okay, so whenever there is a difference in characteristics from offsprings to that of their parents is called as variations. Variations can come in two types. One is called as hereditary variations, other one is called as environmental variations, guys. So what is the difference between these two? So hereditary variations, let's say that. So we have a common denominator for how all the human beings have to appear like. Okay. Whenever they pick up a variation during the course of their living, okay, and these characteristics that they have picked up can be transferred from one generation to another generations. Okay, so these genetic variations are called as hereditary. Okay, they can be inherited. For example, even though it is a negative thing to talk about hereditary variations, let's say that a population gets exposed to some nuclear material or radiations. So when they get exposed to radiations, they have their genetics damaged. And when they give birth to the young ones also express the cancerous traits. So they picked up these variations during the course of lifetime, but these variations became inheritable. So they are called as heritable variations. Got it right? So whenever you can pass on to the characteristics, and these characteristics, whenever you pick up a characteristic, and then you can pass on to these characteristics in the form of heredity, they are called as hereditary variations. Okay. So what is environmental variations? Environmental variations are quite localized to a particular lifespan. For example, let's say that I am living in South India, and then I decided that at some phase of my life that I have to migrate towards the Uttarakhand or Himalayas. I want to go to Himachal Pradesh. Now, what happens here is I have to live in a high altitude region. So, for the first few months or few years, my body will have to learn to adapt to live in higher altitude environments where there is very little oxygen. After the few years, what will happen is my own body develops a specific type of a gene or ability where my body can function very normally in a hypoxic conditions where there is very limited oxygen. So this variation was picked up by the environmental factors. Now, when I give birth to the young one, that doesn't mean that he will also be having the ability to live in the higher altitude. He will carry the exact gene as a South Indian gene, and his body has to adapt to that environmental conditions, and he will adapt to his own pace. He will adapt to his own lifestyle. You get my point? So this is called as environmental variations or non-heritable variations. Another example we can take for non-heritable is, let's take that we have mice. And then what I do is I will cut the tails of the parents of the mice. So this is the variation they acquired through the course of lifetime, correct, right? Now what happens? They give birth, the male and the female mice, they give birth to the young pups, okay, mice pups. So when they give birth to the mice pups, what will happen here? Do the mice pups also lose their tail, or will they be born with the tail? They will be born with the tail, isn't it? So these variations, whenever the environmental factors causes change in their morphology or physiology or biochemical ways, but cannot be inherited from one generation to another generation, is called as environmental variations. And we cannot address genetics without really talking about Gregor Mendel. So he's a versatile genius, a man who is quite far ahead of the time that he lived in. Okay, so a short story about Mendel is he lived from 1822 to 1884 in Austria. So he's a monk basically. So he was very famous for his hybridization experiments with respect to pea plants. So now what happened is his work was published, okay, in something called as translation of the characteristics, okay, sorry, translation of the characteristics in the Natural History of Society, Brunn. So he publishes his work there. But his work was not quite recognized, and it was not given the appropriate, what do you tell, recognition that it deserved back in the days. Okay, so he's also famously known as the father of genetics. There are quite reasons why he got such a popular title to himself. So no, he was a very poor peasant. That means his parents were farmers and they were not economically strong. So they used to work in the fields. So very poor family. So now what happens is he had to stop his education in a very schooling age because he was not able to support. That's why he suspended his education because of poverty and he joined church. In 1843, he joined the church as a monk. So now what happened is his interest, even though he's a religious, he had to be quite philosophical, but his interest to science did not stop there. So what happened is in 1847, what he did was he became the head of the monastery, okay, in the Austria, which is presently in the Czechoslovak, okay, so in Brno. So now in 1851, just shortly 5 years later after became the head, now he picks up his education again. There he goes to the University of Vienna, okay, to study the natural history of and mathematics for 2 more years. So after he exposes himself to this education, now his interest in academics rises and he starts becoming a tutor or a teacher. So when he starts working as a teacher, finally what happens is, where does he become a teacher? In natural history and mathematics. So that's what he teaches between 1856 to 1865. Even though all this information is not really, very important for us to in the examination perspective, but just to appreciate the story and appreciate how much of what kind of an inspiration is Gregor Mendel. So we're just understanding his lifestyle. Okay. So during the period where he was teaching, for the course of 7 years, he started working and designing the breeding experiments. So why I call Gregor Mendel as a versatile genius is, even though we have proof that we do also have proof, and we also can see the insights in the past where other peoples also have attempted some of the hybridization techniques like emasculation and bagging, what we study in sexual reproduction in plants. So, but very well recorded and documented forms is very well visible with respect to Mendelian genetics. So he started formulating his methods where he started designing breeding experiments with respect to the pea plants. Now, when during his breeding experiments, the results were analyzed quite mathematically. So now biology and mathematics is quite contrasting when it comes to the matter of interest. Even though they have the interlinks, very deep interlinks, and they have to be studied hand in hand to for better understanding and more statistical analysis. Right now we appreciate that. But back in the days, it was quite strange for anybody who could bring mathematics into biology. You can understand the relation here, correct, right? Now, what happened? He interpreted all this data mathematically. Now, in 1865, what happened? He published all his results. What I discussed, translation of the characteristics, a natural history of society of Brunn. So, see, look here. In 1865, he publishes. Until 1900s, okay, we don't have any of the appreciation or any of the recognition of the Mendelian's work. It's not about telling that Mendel you have done a great job, but the thing is, Mendel's genetics, if it was understood back then, what he was trying to tell, probably the genetic back in the days, we were like scientists were like the superstars, like the movie stars right now. Probably it would have revelated even more for the discovery of more and more genetics would be unveiled for us in a very short period of time. Anyways, in 1900s, what happened is the same Mendel's work was rediscovered by the three scientists. It's very, very important that you remember their names. Who are those? One is Hugo de Vries, who is from Holland. And then you have Franz Corens, who is from Germany. And then you have Erich Schamark, who is from Austria. So what they did was they reworked the Mendelian's genetics in different plants, not just pea plant. They also worked with pea plants. So, okay, apart from pea plants, for example, you have the 4 o'clock plant. Okay, 4 o'clock plant was worked by the Hugo de Vries. So different plant species they take, and then they try to mimic what Mendel was trying to tell. So they understood what Mendelian published through their experiments. So they revisited the experiments. They did the same way like the Mendel did, and they analyzed this data, and then they translated what Mendel was trying to tell to the current understanding. So when they told that Mendel was trying to really, really mathematically be accurate, okay, about how the characteristics are transferred from one generation to another generations, until then, people did not realize what a grave mistake they did in neglecting the Mendelian data. Why? Why did Mendel's work was completely buried, under the rock? The reason is very, very simple. He had to go publish and talk about his results. Even though it's a scientific community, it is first coming through the church of science. So when you go and publish your work, especially the transfer of characteristics in the church, first thing you're making two mistakes. One is you're talking about something quite religious. So when a person is born or an individual is born, we relate it quite spiritually with the God's creation. So now bringing the mathematical data and telling that it is something called as factors. So these are transferred from this ratio to that ratio. And this is how the characteristics get separated. This is called as a dominant character. This is called as a recessive character. People were not ready to believe. Okay. And moreover, sexual reproduction was not quite yet been very well understood back in the days. This is the second thing. And the third one is the mathematical data. That becomes another bigger problem. And finally, what will happen? Completely neglected, and people believe that it is a coincidental characteristics. That, for example, the question Mendel. See, you are telling that the factors are dominant and recessive. So dominant get expressed, recessive get suppressed. Now tell me, I have a father and I have a mother, and then I am born and I'm completely different. Neither I look like my father nor I look like my mother. I express something else. Now, where is your experiments? It's wrong completely. So Mendel's work was based only upon, okay, I mean, like, what do you tell? Contrasting characteristics, not monohybrid, contrasting characteristics. He could not explain the non-contrasting characteristics. What are non-contrasting characteristics? Something which is not opposite, tall, short. So this is called as contrasting. If I have tall, short, and medium, this is called as non-contrasting characteristics. So whenever there was a situation of non-contrasting characteristics, Mendel became mute. He was not able to explain how. Okay. So anyways, so this was the failure of why the Mendel's work was not quite appreciated back in the days, and it was neglected. Okay. Chalo. Are you guys able to follow? Can you just give me a thumbs up or yes, no, or is everybody able to listen to the class about the voice audibility and everything? Please confirm. Yes, yes. Okay, I can continue, right? Chalo. Sorry about it. Yeah, here we are. So now let's say that why Mendel's when we see Mendel's work is very, very profound. Why? Because first thing is he picks up the contrasting characteristics. So the reasons why he picked up the pea plant, we have the reasons. I will come to that. So one of the very, very, I don't know whether it is a coincidental or it is a very well thought decision that Mendel took. Any other organism in between he would have chosen, Mendel's work wouldn't have finished in 7 years. He would have taken forever to understand how the genetics work. For some reason, he picked up contrasting characteristics. You don't understand the importance of contrasting characteristics. Let me give you an example. Now imagine Mendel picked up two puppies. One is a black puppy, other one is a white puppy, a dog. Okay. Now he will try to cross these dogs, male dog, white and black. Now imagine the next generation. What would be the colors of the puppies? Can you predict the colors of the puppies? Would it be black? Would it be white? Would it be black and white mix? Would it be brown? Would it be gray? So there are so many possibilities, right? Intermediate characteristics are so high when it comes to non-contrasting characteristics. That's why we have to regard Mendel's for his choice with the pea plants. So what are the characteristics that he chose to do his experiments on? So there were like 7 characteristics. What are those? Seed shape, seed color, seed shape, seed color. Then you have pod shape, pod color, flower location, plant size. So we know that all these characteristics, I think you already are aware of it. So in color, coming to seed shape, we have round and wrinkled. All the above ones are the dominant ones. All the below ones are the recessive ones. Okay, we'll understand what is dominant and what is recessive in the coming slide. So, okay, round, wrinkled, yellow, green, inflated, constricted. I think you know the meaning of what is inflated, in like a balloon, inflated. Constricted means when you take out the air, that is called as constricted. Okay, next one you have the color of the pod, that is green pod, yellow pod, purple flower, white flower, axial, terminal. Then you have tall and then you have dwarf. So these are the contrasting characters, 7 contrasting characteristics that the Mendel chose to work upon. Okay. Now, before we understand the Mendelian genetics, before we understand how he performed his experiments, few of the terminologies that we have to be aware about. Okay, let's revisit the terminologies in genetics. Now, Mendel talks about something called as a factor. Now, many of us confuse when a teacher says Mendelian factors. Okay, when two factors, okay, of the same character, okay, are crossed with each other, one of the factor gets expressed, other factors will not get expressed. Now people are confused. What are they talking about? Okay. Now when the two genes coding for the same character, okay, now what will happen? Are expressed. One of the genes get expressed, other genes get repressed. Now we should not get confused between these two terms called as factors or genes. Factor is nothing but genes. Genes is nothing but factor. Back in the days, Mendel referred it to as factors. Okay, and later on, it was understood that these are the genes, a physical part that is present in the chromosome. Okay, so what is that? They are the functional unit of heredity. So we know that how do they become functional? Any part of your DNA or any part of your chromosome that expresses itself in the form of characteristics, those are called as genes. Okay, so that is why they're called as functional unit of heredity, and they're responsible for expression of characteristics in the progeny. Got it right? So where are these genes located? They are located in something called as locus. For example, you have a chromosome. You understand my point? So you can see that, let's say that you have, this is the position. So the genes are located in the same locus of a homologous chromosome. This is a statement. What is homologous chromosome? Let's say that we have 23 pairs of chromosomes, human beings, 23 pairs, right? So that means how many? We have total 46 chromosomes in a diploid cell. So what do we say? How is it arranged? 1, 1, 2, 2, 3, 3, 4, 4, 5, 5. Like that, it all the ways goes to 22, 22, and then we have 23, 23. Correct? No, people agree? So this is how our chromosomal arrangement is there. Now, if I take only 1, 1, that is what is it called as? It is called as homologous chromosomes. If I take 2, 2, it is called as homologous pair of chromosomes. If I compare between 2 and 3, what are they called as? They are called as non-homologous chromosomes. Okay, you get my point. Okay, so now what happens if I talk about the location of a gene or an allele? Okay, I refer it to the loci. So if the genes are located on the same loci of a homologous chromosome, means what? That means if the gene is present in one chromosome like this here, it is the same reciprocation. It is present on the same location in the other chromosome as well. But are they non-homologous? No, they are homologous chromosomes. Now you understand what is the difference, what is the meaning of homologous. Correct, no? Agree? You understand? Chalo. Next one is, what is an allele? Okay. So allele is also, for example, it is an alternate term that we use for the genes of contrasting characteristics. Okay. So alleles actually are paired factors. Like genes are also paired, alleles are also paired. So if I have to express how the genes actually segregate, how the genes actually get from one generation to another generation, how they separate. So I talk about alleles. Alleles, for example, see, tall is a character, correct? No, height is a character, tall or dwarf. Now, if I express the genes, I will call it as tall genes. But if I express the same genes in the form of alleles, I take it as pairs. You get my points. So I take this is one allele, this is another allele for the same character. So why am I taking two? For example, see here, let's say that I have chromosome 1 and then I have another chromosome 1 and then I have chromosome 2, chromosome 2, chromosome 3, likewise, correct, right? Now, if I take that this is an allele, height of a gene, height of a plant is located on chromosome 1, let's take that. So this gene is located where? On chromosome 1. So where should it be located? Another gene. It should be located, see, another chromosome also should be there in the other chromosome, homologous chromosome, right? So let me make you clear. If I have one gene in one chromosome, another homologous chromosome also should carry the same gene, right? So that's why we always represent alleles in pairs. Okay, in a diploid condition, the chromosomes are double. Okay, so 1, 1, 2, 2. Homologous pairs are like double, right? 1, 1, 2, 2, like that. So if I have a character that is coding for, if I have a gene that is coding for a particular character on chromosome 1, it is also present in the homologous pair. That is another one also. So that is why we represent genes in the form of alleles to understand how their characteristics are there, whether they are contrasting characteristics, whether they are similar characteristics. Okay, so etc, etc. Okay, but they are present on the identical locus. So identical locus means, I told you already, if I have an allele present here in the chromosome 1 at this location, the same gene will be the same gene will be located in the same locus of a different chromosome. You understand? Now, are the genes same? Maybe, may not be. For example, this can be capital T gene, this can be small t gene. You get my point? Or this can be capital T, this can be capital T. This can be small t, this can also be small t. So the gene for a character is same. What kind of a gene might differ? Okay, it can vary. So this is about alleles. Okay, next one, what is a phenotype? Okay, pheno means what? Physical characteristics. If I talk about a cross or if I talk about a characteristics, so let's say, guys, if I ask you to look at an animal, I will ask you to describe it. So now you will tell, what you tell? You tell, sir, this is how it looks like. It has a trunk, it has a tail. The tail looks like this. The skin color is like this. The eye color is like this. The appearance is like this. So any noticeable characteristics physically, that is, when you look at it, you are able to tell it by its physical characteristics or physical appearance. So pheno, whenever you see something and you can analyze through your naked eyes, okay, or examine the physical characteristics, this is called as phenotypic characteristics. Okay, so external appearance or morphe, okay, so of an organism. How does the characteristics come? The physical characteristics. It's again because of the genes, again because of the environmental factors. So these are called as phenotypic characteristics. Now, what is a genotype? Something which you cannot see. Maybe you can expect, you can predict to a certain extent what kind of a gene a particular organism might have, but you can't be 100% sure. For example, we might have seen in the dihybrid, in a monohybrid cross, in a heterozygous condition, if I have capital T and small t as a condition, what is the phenotype of a plant? The phenotype of a plant is tall. So if you don't understand this, we'll come to it. If I have a capital T and a small t together in a plant, so when Mendel crossed a tall plant and a dwarf plant, what did he get? He got all of them were tall plants. But did all the tall plants have the same genotype or the genetic character? Was it same, guys? Tell me. Did they have the same genetic nature or did they have little bit difference? What do you say? What is an allele? Okay. Harish, do you still have the doubt? What is allele? Or is it clear? Yes, different. Neha says it's different. Exactly. It should be different. Okay, it's clear. Okay, fine. You get my point, right? So now what happened when we talk about a genotype, we have two different genes, but the phenotype character, you see, it's tall. If I take capital T also, it will express the same phenotypic character, that is tall. So genotype is something you cannot see. You have to analyze through a lot of experimentations. Okay, so there is a methodology. So one of the methodology how to analyze genotype is we just understand how the Mendel conducted his experiments. So you see test cross, you see back cross, you see out cross, then you understand, okay, Mendel was not really insane to just perform crosses. He had some background thought to it. So he wanted to understand, okay, if I have a tall plant, what might be its genetic makeup? So that to understand what is the genes that are hidden, so he had to conduct certain crosses. Okay, so something that is you cannot see, but you can understand through serial breeding, that is called as genotype. Okay, next one is, what is phenotypic ratio? Now, phenotypic ratio, for example, if I have a generation, we have a parental generation and we have a filial one generation, F1 generation, F2 generation, or children we call it as. F1 generation is children, F2 generation is grandchildren. You understand my point? So how do we have here people? So we know that we have parents, okay, give me here 1 second. Through parents, what do we get? Through parents we get offsprings. So we call them F1 generation, children. And through offsprings, what do we get? F2 generation. What do we have? What do we call them? Grandchildren. Correct, right? Now, if I take from parents to the filials, if I take parents and if I cross the parents, and what will be the next generation? How do I tell that? What kind of an outcome did I get? I see the physical characteristics. When Mendel took tall and dwarf and he crossed, what was his result? All of them were tall in the F1 generation. Correct, right? Now, when he go for the F2 generation, when he crossed the tall and the tall, okay, when he took the heterozygous tall and then when he took the another heterozygous tall and he self-crossed, what did he get? He got 3:1 ratio. What is it? 3:1? He got 3 tall and 1 dwarf. How did you get this ratio? You were able to see that in the offsprings, okay, 3 were showing the tall character and one of the plant was dwarf. So you're able to physically examine. So out of the whole population, how many you can group them into tall, how many you can group them into dwarf, how many you can group them into purple, how many you can, or violet or blue, round or wrinkled, green and yellow, inflated or constricted. So any of the characteristics that you can group according to the physical characteristics, okay, physical examination. So this is called as phenotypic ratio. Got it? Next one is genotypic ratio. What is genotypic ratio? Got it. So now you see here, even though in F2 generation का phenotypic ratio, I know that it might be a little weird for you because what am I talking about this monohybrid cross, what is tall, what is dwarf? Don't worry, we'll come across, we will revisit the topic. Let's say that in F2 generation, okay, what was the phenotypic ratio? I said that it is 3:1. Got it? Now, if I see the F2 generation genotypic ratio, what will it be? It will be 1:2:1. How did I get this? I got it because one of the plant was capital T, one capital T, small t, capital T, small t, small t, small t. So three of them were expressing the tall character, that is why it is 3. One of them were expressing small t and small t, that is why it is 1. You get my point? Sorry. Oh, you get my point? So anything in a population, how many plants carry a same genotypic or genetic makeup? Two of the plants were carrying the same genetic makeup. Capital T, small t were two of them. That's why we give them 2. So capital T was only 1. That's why we give them 1. And small t was only 1. That's why we give them 1. So when I talk about a genotypic ratio, I represent it by the common genetic makeup in a population. Okay, or a different genetic makeup in a population. So in phenotypic ratio, physically how much of my population I can divide into groups. Okay, in genotypic, genetically how many groups I can make, how many similar groups I can make with respect to the genetic makeup. So this is called as genotypic ratio and this is called as phenotypic ratio. Now it's a different chromosome. See, is all 7 traits genes were present on a same chromosome or different? Definitely different chromosome. Okay, so now take it as an assignment. Just see the how many number of genes, how many number of chromosomes do the pea plant have, then you will get your answer. Okay, then you'll get your answer. Okay, so as simple as that. Now what is homozygosity and what is heterozygosity? So terminologies are very simple. Okay, and but you just complicate it just because it's introduced to you very rapidly, okay, suddenly. So that's when it becomes a little tough. Okay, let's see. What is homozygous? Okay, zygous. Okay, now homo. Now if you see, when I represent a particular characteristic, I always represent in the form of pairs. I already told you why I should represent a characteristic in the form of a pair. Now, this pair representation, I'm not doing it because I like it. It is because it is representing something more. So if I represent a characteristics, let's say that my character is the height of a plant. Now to represent it, what do? I represent it, okay, height of a plant through some letters. What is my letter? If I have to represent the tallness of a plant, I will take it as capital T and capital T. If I have to represent a dwarf nature, I will represent it as small t and small t. Why I chose capital T, why I chose small t is secondary. Okay, why I put it in the pairs, I told you the reason. Okay, for a character to come into existence, it has to be present on a chromosome in the form of a gene. And to be present on the chromosome in the form of a gene, it cannot be one, because in many of the most of the organisms, right, in all the eukaryotic higher organisms, what we have is, we have all these genes, okay, all these chromosomes which are in diploid nature. If you see a human being, we have 46 chromosomes. Okay, in what cells do we have 46 chromosomes? Can you just tell me where all where all we can find the 46 chromosomes? Can you just tell me, guys? Sorry, can you guys tell me? Can you please tell me? Okay. Uh, you see, Sneha says it is gametic cells. So in gametes, you say that it is diploid. Is it right? No. In which cells? Somatic cells. Exactly. Vegetative cells. Okay, we have two types also. One is vegetative cells, other one is germinal cells. Okay, so now when you look at the vegetative cells or the somatic cells, every cell that is in your body, majority of them, your hair, your skin, your lungs, your liver, your heart, your kidney, all of them are made up of what? Including your spermatogonia and the oogonia. Correct, right? They are all what? They are all diploid in nature. What is haploid? The germ cells are haploid. What are the germ cells? In human beings, it is sperm and ova, or in majority of the animals, mammals, if we consider it, sperm and ova. If you look at your plant species, we call it as pollen and ova, or the egg cells. They are all what? They are haploid. Now, my question is, when I talk about a diploid cells, when I talk about somatic cells, why did we separate as somatic and germinal? We know that these things are quite different in their chromosome nature. Their cell nature, cellular nature is quite different. So when you look at the somatic cells, so the regular cells, they are in diploid form. So when they are in diploid form, you know that for every chromosome there is an alternate pair. So for as I told you, 1 1, 2 2, 3 3, 4 4, like that. So if I have one of the alleles in one chromosome as capital T and in another homologous chromosome also as capital T, they are called as what? Homologous pair. Got it? So let's say another example. If I have in one chromosome as capital T, in another chromosome I have the different allele, that is a small allele, small t, then what do I call? It is called as heterozygous. So whenever the same alleles are there, they are called as homozygous condition. Whenever you have a different alleles, they are called as heterozygous condition. Okay. So non-identical gene pairs which are present in the same non-identical genes which are present, okay, as an allelic pair, they are called as heterozygous. If they're identical alleles, okay, they are called as homozygous. Is it clear? Did you guys understand all the terminologies? Okay. Yes. No. Guys, can we continue? Yes.
Yes, sir.
Yes. Perfect. Chalo. Next, we go for what is a dominant allele, what is a recessive allele. So let's say that if two alleles are existing together, so we know that we can have two conditions. One is similar alleles, other one is dissimilar alleles. So we know that, let's say that I have two alleles which are T together, and I have another pair of allele called as capital T and small t. Now what happens is, here also I get a tall, here also I get tall. Correct, right? In a monohybrid cross, you would have studied about it. So what is a dominant gene? Now look here, whenever the alleles are present in a heterozygous condition, when can I understand the dominance? For example, I am able to differentiate as dominant and recessive when I have two different nature. Correct, right? So one should be short, other one is other one should be tall. Only then I will be able to make I can categorize it whether it is dominant or recessive. If I have a similar, if all of them are tall, how will I? What is the use of me dividing it? There is no point. Correct, right? So I have to have a difference. So now the first point is I have a difference. What is the difference? When the alleles, okay, when the alleles are non, when the alleles are in what condition? Heterozygous condition. It's not homozygous condition. If I have capital T, there is no point in explaining whether it is dominant or recessive. Both are same. The expression is same. Same goes with small t and small t. The expression is same. But if you see here in this condition, alleles are in dissimilar forms or heterozygous forms. So when they are in heterozygous form, one of the alleles will express and the other alleles will not express. So the expressing allele, whichever will show its complete expression of the characteristics, it is called as a dominant allele. Now, when I talk about dominance, there is no intermediate to it. You cannot even differentiate whether it is homozygous or heterozygous. It's as clear as that. So what happened to this recessive allele? Why is it not expressing at all? So the reasons are many. Let's say that there is no protein or there is no function for the recessive allele. It just exists. So only when there is no protein or when there is no hormone produced, with respect to giving the height of the plant, then what will happen to the plant? The plant will become dwarf. Correct, right? So for that to happen, both of them should be in what condition? Small t and small t. So if there is small t and small t, there is no hormone for height. So obviously the plant will become dwarf. So since the small t will not produce any protein at all, just because having one functional capital T, it will compensate. It will compensate completely, just like that of your normal plant, capital T and capital T. That's how the dominant works. That's how the characteristic of a dominance work. So whenever the alleles are in heterozygous condition, one of the alleles will completely express over the other allele. You won't even know that if there is other allele existing or not. So this is called as a condition of dominant. So what is recessive allele? Opposite. Whichever in a heterozygous condition, okay, the allele which does not express at all, they are called as what? Recessive alleles. Okay. When which conditions, guys? Only in heterozygous conditions we can understand this. Okay. Now, what is a hybrid? What is a hybrid? Now, what I did was I take two homozygous characteristics, let's say. What is a homozygous characteristics? You know, you should be aware of the terminology. Homozygous. That means I take similar kinds of alleles in one plant and then I cross it with similar types of alleles in other plant. When I take homozygous parents of two different characteristics, what is the two different characteristics? One is a tall characteristics. Other one is called as a dwarf characteristic. So I have taken two different characteristics, but they're in homozygous condition. Okay, now I cross them. When I cross them, I get all tall plants. But if I look at the genetics of this tall plant, all of them are having capital T. I mean, like some of the plants will have capital T and small t. So when I take the two different parents and I conduct a cross, and the resulting offsprings are called as hybrids. Okay, so they have the characteristics of two different parents. For example, capital T and small t is a hybrid, I can tell. Okay, so this is about what is a hybrid. Sorry. Next one, what is a back cross and what is a test cross? So back cross and test cross, I would be talking about very detailedly as we talk about the monohybrid cross. Is it clear, students? Yeah. Is it fine? Chalo. Now, the thing is, just give me a second. We have come to a certain stage